Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about reaction catalyzed by enzymes. So this is syllabus here, nucleophilic displacement on phosphorus atom, multiple displacement reactions and coupling of ATP cleavage to endergonic process, transfer of sulfate, addition and elimination reaction, enolic intermediate in isomerization reaction, P cleavage, this is not P cleavage, this is the PO cleavage, and condensations, some isomerization and rearrangement reactions, enzyme catalyzed carboxylations and decarboxylation. So this is the syllabus of bioorganic chemistry semester 3. So I will discuss each and every topic in detail. So first start with the nucleophilic displacement on a phosphorus. You know, nucleophilic reagent or group are electron rich species. You all know, and have a tendency to react the atom of a compound or reagent which is electron deficient. Nucleophilic displacement reaction involve displacement of a nucleophile by another nucleophile. You can see here nucleophile plus XY, it gave X nucleophile and Y minus. What does it mean? Nucleophile replaces Y. So nucleophilic catalysis accelerate rate of reaction, and at the end of the catalysis the nucleophilic catalyst is regenerated. So this topic I think clear to you. What is the meaning of nucleophile, what is the meaning of nucleophilic displacement, and these things happen on phosphorus. Now, uncatalyzed reaction and catalyzed reactions. What is the difference between them? You can see here, Rx plus water undergoes solo reactions, it gives ROH, alcohol, and X minus plus H plus. And catalyzed reaction Rx plus Y minus, it gives RY and X minus nucleophile. So Ry plus H2 ROH X minus H plus and Rx H2 ROH X minus H plus. So in covalent catalysis, catalysis have also many types. In covalent catalysis, catalyst takes part during the reaction. Nucleophiles are very effective and versatile catalysts. The enzyme molecule may have three types of nucleophilic groups which work as a catalyst. You can see hydroxyl group of serine CH2OH, sulfahydryl group of cysteine CH2SH, imidazole group of histidine. So some of the nucleophilic substitution reactions on phosphorus atom are as follows. You can see here phosphorus double bond oxygen, single bond oxygen, have charge oxygen. So how this attack on phosphorus and it is in equilibrium. So final products you have here OR and RO, O minus CO. So in this reaction A, covalent catalysis takes place with the formation of phosphoenzyme intermediate compound. Due to nucleophilic attack on phosphorus of the enzyme, the intermediate compound thus formed loses. What loses is clear to in our next slide. Look at here this slide. OR group to give the product, thus OR minus group is displaced by X minus. What happened here? You can look at here the phosphorus double bond oxygen and OR and the F. So these groups OH, serine, En3, it give HF and the product. So reaction B represent the displacement of F minus with ester group. Ester group here, which is the ester group? You know the C double bond OR is the ester group. So here is the ester PO groups. Nerve gas diisopropyl fluorophosphate DFP reacting with the OH group of an enzyme gives, here written differently, gives GIVES, gives HF and orthophosphoryl ester. Now my friends, this type of mechanism of enzyme catalysis is generally seen in the reactions of transferase enzyme, and hexakinase is an important enzyme of this type. So now look at the third. For example, formation of fructose 1-phosphate from fructose in presence of fructokinase. You can see that this is the structure of fructose and fructokinase. You can see how CH2OH is replaced by the OPO phosphate group. So this is the fructose 1-phosphate. Now my friends, I will show you our next slide. You can see here D. Similarly, transfer of phosphoryl group from ATP to glucose is a nucleophilic reaction which takes place in presence of phosphatotransferase enzyme. Look at here, alpha D glucose, here ATP, adenosine triphosphate. I am showing here only the phosphate, and reaction site means reaction occurs on the phosphate, and how alpha D glucose beta phosphate will form an ADP release. Now, multiple displacement reactions and the coupling of ATP cleavage to intergonic process. You know, in ATP, the phosphate group nearest to the ribose is termed as alpha phosphate group, while the other phosphate are labeled as beta and gamma, lambda. You can see here alpha phosphate, beta phosphate, lambda phosphate. You know also adenosine diphosphate and adenosine triphosphate are involved in oxidative phosphorylation. So this is the complete structure of ATP, adenosine triphosphate. Practice with the structure, because this structure, if you know, then you will understand the reactions of this chapter, because every reaction occur on the phosphate and the base and the sugar. So ATP coupled reaction. You know, energy released when ATP is broken down into ADP plus phosphate is transferred to the intergonic reaction through coupling. If you couple the reaction, glucose breakdown and protein synthesis. You can see the glucose, you can see the carbon dioxide, water, heat, and meanwhile this is exergonic, glucose breakdown. And intergonic ATP synthesis, you can see the A and phosphate, phosphate, phosphate. And again, exergonic ATP breakdown forming APP 1-phosphate release in ADP plus heat. And again, amino acid intergonic process, protein synthesis and protein. So this is the schematic diagram you should look at here and try to understand, write many times so that it is conceptually clear to you. Now, you can see here the cycle, charge battery ATP. Just imagine. And H2O release energy, it is converted into ADP and inorganic phosphate. Then dead battery. Again required energy, it is converted into ATP. That's why we are saying adenosine triphosphate is the powerhouse of energy resources. Now, you can look at here ATP, the structure of ATP, and after reaction with the water and energy release, intergonic reaction, intergonic reaction, then inorganic phosphate plus ADP. Now 1 phosphate of inorganic phosphate, we are saying this, is removed from the and forming adenosine diphosphate, and exergonic reactions and inertia, like that reaction is happen in biological process. So ATP and ADP, you can see in this pictorial diagram. In this another pictorial diagram, you can see now it is clear to you how ATP converted into ADP and what is the meaning of exergonic or intergonic process. Again, another pictorial diagram here. You can see adenine, ribose, phosphate groups, release energy, ADP, adenosine diphosphate, absorb energy, release energy, absorb energy, and this process is going continuously. Now, in the cell, the conversion of ADP to ATP is used to store energy, and this energy can become readily available by the conversion of ATP to ATP. And this energy producing system is a cyclic process in the cell. Look at here, ADP plus phosphate, phosphoric acid, 12 kilocalorie per mole, it converted into ATP plus water. And ADP water, it converted into ATP and phosphoric acid plus 12 kilocalorie per mole. So ATP is one of the most important compound in the cell, since its two terminal phosphate groups are linked by the high energy phosphate bond. That is, it has two energy rich bond. It has a high potential for, I will show you in our next slide. You can see here high reaction energy group transfer. Depending on the nature of the bond of the ATP molecule which react, it can transfer following four different types of groups. Transfer of orthophosphate group with the release of ADP, transfer of pyrophosphate group with the release of AMP, transfer of adenosyl monophosphate group with the release of pyrophosphate an activated compound, and transfer of adenosyl group with the release of orthophosphate as well as pyrophosphate. Now look at here. This is the pictorial diagram. Is showing many things. First you look at ATP structure and the reactions C and reactions RCOOH, reactions A, reactions B and reaction C. Then it will clear to you. So it is better you have to write this scheme and try to understand. Here P means phosphate, O you understands what is this. You know the base, you know the triphosphate, you know the adenosine. Now, out of the above four reactions, reaction A is the most common in case the orthophosphate residue is transferred to water. Hydrolysis results. Enzyme catalyzing these reactions are known as adenosine triphosphates, ATPase. Reactions B, transference of a phosphate group occurs rarely. One example is the conversion of ribose 5-phosphate to 5-phosphorybase 1-pyrophosphate PRPP. The reaction C, transference of adenosine monophosphate, is again quite common. You know, my friend, that reaction D, transfer of adenosyl residue, plays a part in the formation of the active methyl groups. So if you practice that reactions A, B, C, then you will understand all this phenomenon and try to understand what is going on. So it is important to note that ATP is not the only reactive triphosphate. Other purine or pyrimidines bases may take the place of adenine in the molecules. The corresponding triphosphate replace ATP in several metabolic reactions. In general, by analogy with the AMP and ADP and ATP, the nucleoside guanosine, uridine, cytidine and ionosine form GMP, GTP, GTP, UMP, UDP, UTP, CMP, CDP, CTP, IMP, IDP and ITP. So don't worry about this abbreviation. This abbreviation is very simple. You just know the ATP, then you will understand everything. Now you can see the ATP in this process become available to drive all those processes which required energy. Actually, the number of examples where ATP is utilized is as great as the number of types of physiological work carried on by the cell. And a summary of the various processes involved in intergonic reaction is represented here. You can see the ATP first, second, third, fourth give the ATP, and then ATP used in the synthesis, muscular contraction, nervous excitations, active transport, intergonic reactions. ATP as a common currency of the bioenergetic. So ATP represents the energy currency of the cell. Now let us discuss one example, synthesis of benzoyl glycine, to show how ATP is used. The synthesis of benzoyl glycine, hippuric acid, does not process spontaneously as written below. You can see the benzoic acid plus glycine, it gives hippuric acid plus water. Actually, the synthesis occurs in the animal's body by expenditure of chemical work. That is, it is intergonic. So, benzoic acid plus ATP give benzoyl adenylate and pyrophosphate enzyme bonds. So, my friends, this slide will more clear to you about your syllabus. You know the benzoic acid coenzyme A, benzoyl coenzyme A, AMP, enzyme bond. So both the above reactions are reversible because most of the biological processes are reversible, because in each case the bond broken and bond formed are energetically equivalent. Now once the benzoyl coenzyme is formed, it reacts with the amino group of glycine to form hippuric acid. Now benzoyl coenzyme A plus glycine give benzoyl glycine plus coenzyme A, and this reaction is irreversible and take place only in the right side, because the energy level of the thioester linkage in benzoyl coenzyme A is considerably above that of a peptide or amide in benzoyl glycine. So you have to write all amino acid their structure so that you will understand what is going on in biological process, including a structure of ATP. Now my friend, in other examples where ATP, adenosine triphosphate, is utilized, the same general pattern is followed. Direct reactions between ATP and substrates occurs, work is performed, and inorganic phosphate or pyrophosphate is produced from the original high energy phosphate bond. The same mechanism is believed to occur in mechanical as in muscles contractions, osmotic as in the secretions, absorption and kidney functions, and electrical as in nervous impulses work where ATP is utilized. So my friends, transfer of phosphate, addition and elimination reactions. You know the addition reaction, double bond breakdown in the single bond. These reactions are given by the unsaturated compound in which the reactant adds at the double bonds and resulting in a saturated compounds. Now in biochemical activates, such type of reaction are common. For example, the reaction which takes place in the citric acid cycle, fumarase, you can see the malates. Here what will happen, fumarate, trans and malate. So transfer of sulfate groups, this is also very important. Transfer of sulfate group from PAPs, PAPS to the phenol, alcohol or amine is carried out with the help of enzyme sulfotransferase or sulfokinase. You can see the PAPs plus RZH, it give RZSO3H and 3-phosphoadenosine 5-phosphate PAP. So where Z is equal to O or NH. So sulfation means the second step here is the transfer of sulfate group from the coenzyme PABs to accept a drug by nucleophilic attack. You can see the drug contains OH functional groups. You have the PABs, adenosine, sugar and phosphate. So 1-phosphate drug sulfate conjugate and the PAB. So this is sulfation. So ATP sulfate, ATP sulfurylase, APS and PPI for pyrophosphatase to inorganic phosphate, APS reductase, SCO3 minus, S3O6 minus, H2O3 minus, and finally bisulfite reductase, you will get the S2 minus. So you can see the in biological process what's going on, SO3 minus, H2 minus how convert, and again APS, AMP, you can see the other changing ATP to ADP. So a lot of information it happen when electron transfer chain occurs. So this is an addition reaction which has a specific dimension, as the trans isomers of unsaturated dicarboxylic acid fumarate adds a water molecules at the double bond to give the L isomers alpha hydroxy dicarboxylic acid malleate. The enzyme fumarate does not interact with as isomers of an unsaturated acid and demallate. The stereochemical study using deuterated water indicate that the mechanism involved transaddition of D plus and OD minus at the double bond catalyzed by the enzyme. You can see the transaddition of D2O due to a deuterated form of water. Now similarly, cis-aconitic acid. Aconitic acid, which is an intermediate compound with the enzyme aconitase, add a water molecule to give citric and isocitric acid. On one side citric acid is formed and on the other side isocitric acid. So isotopic studies replacing hydrogen with deuterium in the substrate indicate that hydrogen and OH add trans at the double bonds. So my friend, it is little bit heavy, but if you try several times you will understand citrate. First you try to write the structure several times so that it will be in your mind what reaction occurs here. You can see the citrates, cis-aconitic acid and isocitrates. So look at here, the stereospecific transaddition of water may be represented as follows. Behind the plane and in front of plane you can see isocitric acid. So elimination reaction, you know, elimination reaction are just opposite to addition reaction. In addition reaction, unsaturated compound are converted or converted into the saturated compounds, while in elimination reaction saturated compounds are converted into unsaturated compound due to intramolecular elimination of water, HCl and ammonia. Example, in citric acid, cyclic succinic acid is oxidized into fumaric acid in presence of succinic dehydrogenase enzymes. So my friends, you can see FAD plus and the molecules you can see contain COOH groups is converted into FADH2. Where is the two hydrogen coming? So formation of double bonds. This is because I told you this is happen in the biological process. So this is example 1,2 or beta elimination reaction. In this reaction, succinate hydrogenase enzyme trans elimination 2 hydrogen gave fumarate. Similarly, in close catalyzed glycolysis also involve elimination reactions. So my friends, you can see 2-phosphoglycerylate, enolase, phosphoric acid pyruvate. In this reaction, the unsaturated product is obtained due to elimination of a water molecule from the substrates. Now I will talk enolic intermediates, isomerization reactions, PO cleavage and condensations. You know, condensations, isomerization and rearrangement reaction play important part in metabolism phenomenon. The isomerism is the reaction in which one isomer is changed into the another. In biochemical reaction, these transformations takes place in two ways. By changing a position of double bond with the intermediate chain in the position of two hydrogen atoms. And aldose-ketose isomerism is an important example of intermolecular shifting of hydrogen atom. My friend, interconversion of dihydroxyacetone phosphate into D-glyceraldehyde 3-phosphate, and this interconversion takes place in the presence of triphosphate isomerase. Now you can see here the phosphate, you can see how B minus approaching H plus, and here removal of reaction occurs at the breaking of the PO bonds. So you can see here PO cleavage 30%. You can see here restricted enzyme tools to cut DNA. Here again breaking of the PO bond, phosphodiester cleavage by restricted enzyme. So my friends, it is little bit heavy, but if you practice these molecules, then you will find with the help of general chemistry actually what is going on here. Look at here how it is cleaving the pure bonds. Now, similarly, the reversible reaction which takes place in the presence of glucose phosphate isomerase, you can see dihydroxyacetone phosphate and glyceraldehyde 3 phosphates. So, fructose 6 phosphate to glucose 6 phosphate. In this reaction, indiol is the intermediate compound. You can see the diol. Now, you can see the examples. It is better you write it down in your notebook and see on arrow how OH bond breaking, H plus removing, DH bond breaking, ring breaking, and then further formation of ring. Now, these reactions involve conversion of C1 carbonyl group of glucose into C2 carbonyl group of fructose. In next step, OH groups attached to the C1 of fructose is easily phosphorylized to gives 1,6-diphosphate. You know, my friend, intermolecular reaction of functional group is a comparatively less frequent in metabolic process. However, in metabolism of glucose, conversion of beta-D-glucose 6-phosphate into beta-D-glucose 1-phosphate in presence of phosphoglucomerase is an important example of the phenomenon. You know, in this isomerization reaction, phosphate group present on carbon 6 is shifted to carbon 1. That is very important point here. Now look at here, beta-D-glucose B-phosphate to phosphoglycomutase presents, you will get the 1 beta-D-glucose 1-phosphate. So you can see the pentose phosphate also shows different types of isomerizations. Now the epimerase enzyme making change in the arrangement at the carbon 3 gives the epimer. Hence, the process is known as epimerization, a special type of isomerism you have read in carbohydrate chemistry. Now, you can see the ribose phosphate indiol, ribose 5-phosphate. In the reaction, a ketose is converted into aldose. So, condensation reactions. Condensation is the process of combination of two or more molecules with the elimination of water or ammonia molecule to give compound of higher molecular weight. The simplest example of the process is condensations of two molecules of acetaldehyde in presence of dilute alkali to give aldol condensations. Condensations, you know, during metabolic process of biomolecules, generally aldol and retroaldol reactions are seen frequently. For example, the citrate synthesis catalyzed reaction in the first step of citric acid cycle. The first intermediate compound citric acid cycle is formed due to condensations of oxaloacetic acid and acetyl coenzyme A. Now look at here, acetyl coenzyme A. Focus on the structure. I am trying to give the heart of the chapters, because this is little bit hard, but if you practice then it will be very easy to like the general chemistry. So oxaloacetic acid, then aldol, then coenzyme SH, you can see the succinic acid. So my friends, in this reaction aldol condensation takes place when hydrogen atom of methyl group in acetyl coenzyme A combined with the carbonyl composition of oxaloacetoacetate. Aldol is the intermediate compounds. So similarly, when D-glyceraldehyde 3-phosphate and dihydroxyacetone phosphate condense to give alpha D-fructose 1,2-diphosphate, in the front side aldol condensations and in the back side retroaldol condensations takes place. The enzyme in this reaction is aldols. Now look at here the scheme. Glyceraldehyde 3-phosphate dihydroxyacetone phosphate and forming alpha D fructose 1,2-diphosphate. Now carboxylation and decarboxylation are reversible reactions. In the metabolic process, both these reactions are seen frequently. The example, formation of oxaloacetic acid from pyruvate in presence of an enzymes involve carboxylation. Pyruvate react with carbon dioxide plus ATP and water in presence of manganese 2 plus oxaloacetate ADP plus inorganic phosphates. So my friends, now you can see here, this is an important reaction which is catalyzed by pyruvate or pyruvic acid, and hydroxy acid takes place in different metabolic process. Hence, the decarboxylation reaction is quite common in this process. For example, formation of acetaldehyde from pyruvate in the presence of pyruvate decarboxylate enzyme present in each cell. You can see acetaldehyde formations with the acetone groups. So, next topic is enzyme catalyzed carboxylation and decarboxylation. Similarly, in citric acid cycle, the first step is the formation of oxalosuccinic acid as an intermediate compound due to dehydrogenation of isocitrate, with being unstable readily decarboxylate to give the product. Alpha-ketoglutarate, this is an example of oxidative decarboxylation which takes place in 2 steps. Look at here. I have already shown you, but again you can see NAD plus converted NADH dehydrogenase and oxalosuccinic acid and alpha-ketoglutarate. So, some of the more important biochemical reactions catalyzed by enzyme include nucleophilic displacement reactions on phosphorus atom like ATP cleavage to intergonic process, additions and elimination reaction, condensation reaction, isomerization rearrangement reaction, and carboxylation and decarboxylations. This is your syllabus. So, I will give you one examples complete. Chymotrypsin, this is one enzyme, chymotrypsin. So, what is the function of chymotrypsin? Look at here. Chymotrypsin is an important proteolytic enzyme. It is a protein hydrolyzing enzyme. Like trypsin, chymotrypsin is also secreted as an inactive proenzyme, chymotrypsinogen, which is activated in the intestine by trypsin but not by enterokinase. Its optimum pH is between 8 to 9. It acts is very important same way as trypsin, which preferentially attack on peptide linkage involving the carboxyl groups of trypsin and phenylalanine. You know, moreover, like remains the pepsid, it has a powerful milk clotting action. The end products of chymotrypsin hydrolysis are a smaller polypeptide and free amino acid, trypsin hydrolysis residue of lysine and arginine, while the chymotrypsin hydrolysis residue only of aromatic amino acid. So look at here the scheme: lysine and tyrosine, a specificity of proteolytic enzymes. Now you can see the activity of chymotrypsin is supposed to be due to position of histidine 57 and serine 195 residue near to one another on the active center, and second, covalent acyl enzymes intermediate compound whose hydroxyl group of serine 195 takes place in the reaction. The mechanism of hydrolysis of peptide bond by chymotrypsin is explained as follows. Imidazole group of histidine 57 of chymotrypsin enzyme act as a general base. Now look at here the second very important point for you. Hydrogen atom of OH group of serine 195 residue of chymotrypsin and nitrogen atom of imidazole ring of histidine 57 link through the hydrogen bond resulting in transfer of the hydroxyl proton to nitrogen of histidine 57. Now most important point here, in presence of a base, hydroxyl group of serine 195 undergoes nucleophilic attack on carboxylic carbon of amino acyl group of the substrate and formyl acyl chymotrypsin adduct. In the second step of displacement, serine 195 acyl group act as a donor and water alcohol amino acid amine behaves an acceptor. The last slide which I am going to talk with you is this. So this is my last slide for you. Out of many mechanisms put forward for explaining activity of chymotrypsin, on dipeptide, and this base catalyzed nucleophilic displacement mechanism is an important and cites an example of covalent catalase. So my friends, I hope this video will clear to you. Practice with examples and do preparation of your exams, and thank for watching. Wish you success in your exams.